on AAV production and HEK293 cells. Isso, would you like me to advance your slides, or would you like to advance your own slide?
Yes, if you can do that, please.
Absolutely.
So, yeah, thank you. Hi, everybody. As everybody said before, there are jobs in this market, especially now in the new area, in cell and gene therapy. Yesterday, there was a CEO of a company here in Massachusetts. He came here and he wants 300 workers now, and we produced 300 in 5 years. So that means we have to find a new way to train the people. So training is needed. So as a part of the Nimble project, so we decided to create a protocol so that people can use it as a training. Even industry has also expressed the interest to come. Once we put something in place, they will use it for training purposes before the employees work on cells that are gonna go to patients. So, so that's what we here at Quincy College, we work on AAV2. So we call it AAV2 biomanufacturing. So we produce AAV particles in HEK293 cells. The reason we choose AAV2 is because first, it's the best known among the type of AAVs. And it's easy to get the raw material as opposed to Jim and Maggie, and they have a hard time getting the material to work with the cell line. So this one is relatively easy to get the material needed. So, and also the safety level is relatively low as long as you follow the regulation. So what we knew to start, we didn't know anything about AAV manufacturing. The only thing we knew is aseptic techniques, and we also know how to work with CHO cells. So we do that here, but not the viruses. So this was a brand new thing for us. Next slide, please. Yeah, so that's, as you can see, the NIH recommend that working with AAV is a biosafety level 2. So here, according to the companies that sell AAV and work with AAV, the recommendations are if you use a biosafety cabinet class 2, it's relatively safe. And alcohol is not effective for disinfection, but 10% bleach is the one to use. So that's what we've been doing here. And we can autoclave the biohazard before even putting the biohazard waste. So that's what we've been doing. Next slide, please. So we started with these cells called viral production cells. It's sold by Thermo Fisher. And these cells are designed to produce AAV particles. So we use them to transfect and for transfection. So we bought the cryoval, and when it comes, it's recommended to store it in liquid nitrogen in the vapor. So that's what we did until it's ready to use. And also, Thermo Fisher provides media. So there is, that's in the next slide. So the media, there is production medium and there is a Glutamax feed. So the complete medium gonna be production media with 4 millimolar Glutamax. And that's ready to use. The other good thing with this is that Thermo Fisher gives you all the recommendations, the volumes that are recommended to work with. So you take the cryoval, and we thaw the cryoval. I think that's on the next slide. So yeah, so the cryoval comes from the -150 liquid nitrogen. So here we have a tau star on the right side of the screen. It's just used to de-ice the cryoval. So you push it in, and in less than 2 minutes, you're gonna pop it out when it becomes mostly liquid. If the cryoval is not cold enough, if you take it from -20, it won't work here. It has to be -80 or -150. So we de-ice it and put it directly into the medium. And the Thermo Fisher recommended 30 ml of media. And these cells are trained to grow in suspension. So they have other cells that you can grow in tea flasks, but you have to harvest with trypsin. Since we didn't know anything about this, we wanted to start with what is easy. So here inside the bioseptic cabinets, aseptically, we transfer the entire content of the cryoval into 30 ml of complete medium. And we put it, this is a single-use shake flask. So with a tubing, so if for expansion, can just weld the tubing to a bag and pump it in there, like Maggie was talking about, with a stereo welder. But we, at this stage, we didn't need that. So we just use it as it is. Next slide, please. So after the inoculation, so transfer the entire content of cryoval into the medium and put it in a shake flask, in a shake incubator. And they are recommended to use 8% CO2, not 5% like fuel cell, temperature 37, and the agitation 125 RPM. So, and it's recommended to, they grow not that fast, but they take 4 or 5 days to grow. So that's, we see that here on the next slide, please. So after 3 days, after 4 days, we took a sample and we did a cell count using the automated cell counter, Biorad cell counter. And we can see the viability, the total number of cells. And the good thing is, Thermo Fisher also recommend a cell density that is needed for transfection before we put in the viruses. That's on the next slide. Yeah, so this here is the egg cells before transfection under the microscope. So what we did is we took a small amount, put in a tea flask, and put it under a microscope and look at the shape of the cells. So that tells only the confluency, but it doesn't tell whether the cells are alive or not. The Biorad cell counter tells how many are alive. So this is just for visualization. Next slide, please. So yeah, here is another cell counter. I like this one better because with the Biorad cell counter, when the cell density is very high, you get a zero viability. It shows you zeros because it just can't count as many cells. But this one can count the highest density of cells. So we use this one as well. And so we use this one when we reach a high cell density, we did a cryopreservation. That's on the next slide. So we started one cryoval and we expanded them, had a lot of cells, and we did a cryopreservation. They recommend that we use 10% DMSO and put them in 11 cryovals. Then we have a bunch of cryovals to work with. Since we didn't know so what was going to happen. Actually, at a certain point, we got contamination and we had to use other cryovalves. So that's what happened. Next slide, please. So here is when we had enough cells as recommended by Thermo Fisher with 30 ml of culture. And they also tell you the volumes of plasmid. So we bought the plasmid from another company. That's what Thermo Fisher didn't have. So we had to buy the 3 plasmid from another company called Cell Biolab. And Thermo Fisher give you the transfection reagents, and they give you the recommended volumes for 30 ml of a culture. So we can scale up if you have a larger volume, scale down if you have smaller volume. So that was very helpful. Next slide, please. Yes, so these are the transfection reagents. Thermo Fisher sells these as well. They just don't sell the plasma. So we had to talk to another company, actually a bunch of them, until we found one that has the plasmid. That's on the next slide. So yeah, these are the plasmid called DJ plasmid. So they come in a box, and you have all the 3 plasmid because the process is called triple transfection. That's what Jim was talking about. So you put 3 different plasmid, but you mix them with a buffer, and you just transfer them into the cell culture one time. So, but you have to pre-mix them before in a buffer and transfer them into the shake flask. So these ones, you can also, Jim was talking about plasmid extraction. The good thing, you can actually take these plasmid, put them, transfect E. coli, and scale up, get a bunch of a large volume of plasmid, and you can harvest and cryopreserve the plasmid for the next transfection. So this also can be useful. Next slide, please. Yeah, so this is the cell culture transfected with the 3 plasmid called triple transfection. So the culture goes back to the incubator with 8% CO2, and it's recommended to stay there for 72 hours. That's about 3 days. And then that's what we see on the next slide. Next slide, we continue with that. The kit we buy with from Thermo Fisher comes with also a Lysis buffer. And they tell you the volume needed for a 30 ml of culture. So what we did, just add 3.3 ml of Lysis buffer directly into the shake flask and put it back into the incubator for 1 hour. So that's produces the cell lysis. So the cells birth, and if they took the plasmid, so then they're gonna have the viral particles come out of the cell and gonna be in suspension in the media. So next slide, please. So after the lysis, they stay in the incubator for 1 hour. Then you have broken cell debris media. The viral particles supposed to be in the suspension, liquid on the top. So after centrifugation, at the parameter recommended 10 minutes, 4 degrees Celsius for 13,000 Gs. So then there is a pallet at the bottom, which are broken cells. So we collect the supernatant, and that's where the virus particles supposed to be. When we started, we also, another company sold us some purification kits, some mini columns with a resin. That one we try, it doesn't really work well, it gets clogged. After you put a certain volume, it doesn't go down because you have to do the chromatography by density, so it won't flow through. Finally, we discovered that Thermo Fisher has a resin to bind those particles. So then we tried our resin. That's on the next slide. This one. So the resin is on the left, and but our resin is designed to bind AAV virus. So it also bind all the other type of AAV. Then we had to find a column. That was not easy. We finally found a column from a small company here in Massachusetts. These columns are relatively new. They are not known that well. The G columns are known, but they are big and they are difficult to work with. And for this resin, we just need a small amount of resin. So once we found this column, they called a snap column, and they work very well. You can see top and bottom, they have an adapter. So you can hold the resin in the glass, and you can actually measure the length of the resin. So it's much easier to work with. So we use that part of the column with the resin from Thermo Fisher. Then we went to for purification on the actor. That's on the next slide. So we use Acta Pure, like Maggie was talking about. So the Acta Pure works very well with this. So we inject. So we load the harvest, the lysates. So the suspension of after centrifugation. So that's where the virus are. Then you need 3 buffers, wash buffer and binding buffer and elution buffer. And those buffers are relatively cheap. The elution buffer is a glycine, so amino acid, just 100 millimolar. And the wash buffer is of phosphate, it's a PBS. So we just use the PBS to wash. And also we use the PBS as a binding buffer, wash buffer and binding buffer. It's also recommended to use sodium chloride, 1 molar sodium chloride to wash after the binding. So you're gonna see on the screen, we can't see, it's not clear, but you find out that on the screen, you actually see the impurities when you wash with the binding with the PBS, you're gonna see impurity comes out. But when you use a 1 molar sodium chloride, you see extra impurities coming out. Then finally, after that, we use the loading, the elution buffer. And you can see the DNA, the AAV particle come out, and we collected them. So that's what we have done. So we have collected the particles after purification. And we're working now on the qPCR, the ELISA, and we also working on HPLC to separate the empty capsid from the full capsid. So like Jim was saying, even when you get the capsid, the viruses, some of them will not have the DNA, they are called empty capsid. Some of them gonna have partially the DNA, they're called partial capsid, and some of them gonna be full. So for patient treatment, the ideal is to have the full capsid. That's what we're gonna actually produce an effect. So, but it's not easy to separate them. We haven't been successful so far. There is a column that we found in publications, which we've been using that column, but trying different mobile phases, but it doesn't really separate very well. Hopefully we're gonna be able to present more results during the second presentation in January. So that's where we are for now on that project.
Yeah, so let me add that there are companies that claim they can separate empties from full capsids using chromatography, but I'm not sure I believe them. I'm not sure that they can do it, and so it's a lot to expect us to do it.
I guess Thermo Scientific have an anion exchange column that they claim can separate. Is that the one you tried?
Yes, they do claim it.
Yes, that's the one.
Mm-hmm.
A lot of companies do ultracentrifugation with either cesium chloride or another gradient maker.
But that's not really scalable.
Correct?
Thank you, Izzo. Do we have any questions for Izzo or to Barbara?
We do. One question was the use of adherent versus suspension cells, and is one better than the other for this application or just for industry in general?
I'll add, I see both in industry.
I do too.
Yes, I do too. The only thing is suspension is much easier to work with, especially if you're not used to it. Like us, we just started. We wanted to do something easier. If you use trypsin, there is a risk of killing the cells with trypsin. So we didn't want to do that here.
There was also a question about what cell line you're using from ATCC, because there's an individual who's having a hard time converting their adherent HEK cells to suspension, but we'll provide those details with the lab protocol, correct?
Correct. I believe if you go to Thermo Fisher, they have cell design trained to grow in suspension. That's what we bought. But they have other cell line that can grow in a tea flask, adherent cells, and you have to harvest them with trypsin. So these cells are specifically designed to grow in suspension. I don't know if they can grow. They also can be adherent in a tea flask.
And then we had one more technical question. Is it essential to shake the cells after transfection?
Yes, Thermo Fisher recommended that. Yes.
All right, thank you.
Barbara, I see a couple of questions we didn't get to. One was, do patients get chemotherapy before their autologous cell therapy treatment? And that is yes. So an integral part of that is you hit the patient with chemotherapy, and the way it's been described to me is it opens up the niches that your CAR T can go into. The other question came up is, can you do autologous earlier? And everyone wants to do it as a first line treatment. Currently, the regulatory agency requires a failure in traditional chemotherapy treatment before starting CAR T, but there's no reason not to make that your first line treatment. And that's the hope of everyone.
There was one more question, Jim, I had seen earlier. What's most important for cell line development and clonal stability?
Well, I'll put that a different way. It is important for you to maintain clonal stability. And so that's the reason why you have the master cell bank, working cell bank into the production line system. And so you do maintain clonal stability and you do test for it. And as everyone knows, cell lines, they'll throw out chromosome. If you have them replicate for months and months, they can throw out chromosomes or duplicate chromosomes or do all sorts of weird things. In their karyotype can go all over the place, and you want to avoid that.
Okay, is that any other questions before we wrap up? So I would like to thank the WeBet team for a great presentation. Thank you, guys. And I'd like everyone to look out for an invitation to our Train the Trainer session 2 through Innovate Bio and Sandy Porter. And thank you, Sandy, for for hosting us today and all the work you did ahead of time. So we'll be sending out notification of our second Train the Trainer session, where we'll be talking, as I mentioned earlier, about QC analytics, the regulatory environment, and more hands-on curriculum that we'll have developed and be ready to present to you. So again, thank you everyone for joining.